muy buenas tardes. Les agradecemos el interés y que nos acompañen en este ciclo de conferencias sobre ciencia organizados por el consorcio Universidades por la Ciencia. El ciclo es organizado por Fundación UNAM, el Colegio Nacional y un conjunto de universidades en México y en el extranjero. En las semanas pasadas hemos tenido el privilegio de contar con un grupo muy distinguido de conferencistas sobre diferentes temas y hoy la conferencia está a cargo del doctor Chris Lori de la Universidad de Texas en Austin, del Instituto de Geofísica, y el tema es nuevamente algo muy interesante, que es cómo se recupera la vida después de una extinción masiva que afectó a todo el grupo de los dinosaurios. Esperamos que la disfruten. Nos acompaña en la transmisión el presidente de Fundación UNAM, el licenciado Dionisio Meade, la directora ejecutiva de Fundación UNAM, la licenciada Araceli Rodríguez, también el doctor Gerardo Arizmendi de la Universidad de las Américas en Puebla, y Matthew Willis, el profesor Matthew Willis del Centro Milner de Evolución en la Universidad de Bath, y las organizadoras del ciclo del consorcio, la doctora Araceli Urrutia y el doctor Martín Serrano. Y la presentación está a cargo de la doctora Eligia Pérez Cruz, quien es la coordinadora de las plataformas oceanográficas y en donde se hizo buena parte de esta investigación de la universidad, que nos hará la presentación de nuestro conferencista invitado. Entonces, nuevamente es realmente un placer para nosotros el agradecerles el que nos acompañen en este ciclo de conferencias. Agradecemos a nuestro conferencista invitado por aceptar esta invitación y compartir con nosotros. Esto nos ha permitido llegar a todos ustedes de una manera bastante sencilla y esperamos que sea interesante. Entonces, le pasamos la palabra a nuestro presidente de Fundación UNAM, quien es el organizador del evento, el licenciado Dionisio Meade. Muchas gracias.
Muchas gracias y muy buenas tardes a todos. Nuevamente subrayo el privilegio de participar en este evento formidable que, en coordinación con el Colegio Nacional y con un consorcio que integran 14 universidades, estamos presentando sobre muy distintos temas de investigación científica que hoy nos enlazan a través de distintos países, incluso de distintos continentes, y que nos abre una puerta más al conocimiento. Este consorcio, vale la pena subrayarlo, se formalizó apenas en mayo 20 y fue una respuesta a las nuevas circunstancias adversas que habían derivado de una pandemia que ha puesto al mundo en jaque en temas de salud, que ha subrayado, pues por una parte, la humildad con que debemos enfrentar el conocimiento, pero por otra parte la capacidad de organización, de cooperación y de investigación que el mundo está articulando para tratar de superar esta adversidad. Y parte de la respuesta pues ha sido iniciativas como esta. Esta iniciativa ha sido una respuesta que justamente está permitiendo que, aprovechando las ventajas de la comunicación a través de estas redes, podamos enlazarnos muchas universidades, podamos enlazarnos muchos científicos y podamos enlazar, y ha sido una respuesta que nos ha estimulado mucho, una gran presencia, una gran participación de investigadores, de alumnos de distintas partes, de distintas universidades que están queriendo favorecerse de este nuevo medio de enseñanza, de este nuevo medio de compartir investigación. Se han tocado temas muy diversos, han participado universidades de distintas nacionalidades, inclusive transitando océanos, pero comunicados ya en línea. a través de esta capacidad que nos ofrecen las redes y subrayando el potencial que deriva de esta comunicación. Un potencial formidable, un potencial que suma, un potencial, una capacidad que suma y que permite participar de la capacidad de conocimiento que se está generando en el mundo. Queremos agradecer nuevamente a Jaime Urrutia su decidida participación y sobre todo la gratitud de Fundación UNAM al permitirnos participar en ese evento. Queremos otra vez felicitar a Araceli por haber tenido esta formidable iniciativa y haber encontrado eco en otros participantes del mundo. Y queremos dar también la bienvenida a su participación. No pudo estar con nosotros Martín Willis la semana pasada de la Universidad de Bar. Y saludamos también a Gerardo Arismendi de la Universidad de las Américas, copatrocinador, cofundador de este consorcio. Y damos desde luego la bienvenida a Christopher, que hoy nos va a tocar un tema relevante. Y esto, como digo, nos ilustra el potencial que tenemos de compartir conocimiento, de compartir esfuerzos y de compartir de la mano entre muchas universidades en lo que sentimos que es el cimiento de un nuevo mundo de la enseñanza y de que la nueva realidad ofrece también oportunidades. Muchas gracias a todos, muchas gracias a nombre de Fundación UNAM y escucharemos con el mayor interés la participación de nuestro conferencista el día de hoy. Gracias, Jaime.
Muchísimas gracias, licenciado Dionisio Meade. Es un verdadero honor que Fundación UNAM sea parte de este consorcio. A nombre de todos los participantes en el consorcio de Universidades por la Ciencia, les damos pues la más grande bienvenida a la sesión de hoy. El doctor Martín Serrano de la Universidad de las Américas en Puebla no pudo estar con nosotros, pero en su representación vamos a tener unas palabras del doctor Gerardo Arismendi, también de la Universidad de las Américas Puebla. Apenas la semana pasada fue el organizador de un evento organizado por la Universidad de las Américas y en colaboración con el Consorcio de Universidades por la Ciencia, una excelente presentación del doctor Raúl Rojas. Entonces es un honor tener aquí a Gerardo y pues le doy la palabra. La diapositiva que están viendo contiene las instituciones que forman hasta ahorita parte del Consorcio de Universidades por la Ciencia, que tiene como objetivo promover la colaboración en temas de investigación y la divulgación de la ciencia. Entonces, Gerardo.
Bueno, buenos días, buenas tardes a todos. Es un placer para mí estar aquí representando a la Universidad de las Américas Puebla. Como ya saben, la universidad está muy entusiasmada con este proyecto de Universidades por la Ciencia, que es un consorcio que ha desarrollado muchas pláticas a muy buen nivel, a un excelente nivel, trayendo conferencias de muchas partes del mundo y que ha tenido un alcance increíble. Lo que ha hecho esto un éxito. No me queda más que decir que es un placer y decir que la Universidad de las Américas Puebla orgullosamente colabora con todas estas instituciones y felicidades por esta gran colaboración.
Muchas gracias, Gerardo, por esas amables palabras. Y sí, la universidad ha sido un gran colaborador y ha organizado ya 2 conferencias en sus plataformas de eventos asociados con este consorcio y en ambos casos excelentes ponentes, excelentes eventos. I'm going to switch to English now. So it is my great pleasure to have here with us Professor Matthew Wills. He is the Deputy Director of the Milner Center for Evolution, which is a proud member and a very valued member of this consortium of universities for science. Matthew is an amazing scientist, a very good paleontologist, and he likes to go far beyond what we can learn from looking at one single fossil, and he's interested in the big picture of how life has evolved on Earth. So I am really glad that you could join us today, Matt, and please go ahead.
Well, that's very kind of you, Araceli. Thank you. Well, I'm delighted to welcome you to this seminar by Chris Lowry from the University of Texas. I'd like to say a word in English, perhaps, about the Universities for Science consortium that made the webinar possible and that promoted it. The purpose of the consortium is to bring together institutions and scientists from around the world to promote research collaboration and public engagement in science. Membership is completely free, and it's open to any institution with a mission for international research or science communication. Members are very welcome to share their own activities, which so far have included webinar series and documentaries, but this will soon expand to encompass symposia and jointly taught units. Part of the ethos, I'm told, is to minimize bureaucracy, and that sounds excellent to me. So the consortium currently has over 10 member institutions from Mexico, the USA, and the UK, including, I'm delighted to say, the Milner Center for Evolution, and we're extremely proud and honored to be part of it. So Bath's Milner Center houses PIs from across the University of Bath, have a growing community of researchers at all career points. There are relatively few centres that attempt to bring together all branches and scales of evolutionary biology, and only one, so far as we know, with a joint mission for education and outreach alongside blue skies research and applied research. So please do us. We would be delighted to hear from you. Okay, thank you very much.
Many thanks, Matt. I think those were really nice words, both about the consortium and also the University of Bath. And I know that Alejandro would have been delighted to see you, so he sends his regards. I'm gonna now pass on to Dr. Ligia Perez Cruz. So she is a faculty member of the Institute of Geophysics at the National University in Mexico. And she is also the current director of the, I don't know how to say it in English, but she coordinates both oceanographic ships that the National University of Mexico has. She has a very, very demanding day-to-day job, and also is in charge of elaborating the strategy for how the university will go on to contribute to oceanographic research in the future. So Ligia, over to you.
Thanks, Araceli, for the nice presentation. So you are very kind. Okay, it's my pleasure to present to you Chris Lowry. And it's an amazing young scientist, really. And I had the opportunity to stay with him in an expedition from IODP. Chris is a paleoceanographer, is specializing in foraminiferal micropaleontology. And Chris used a combination of tools such as foraminiferal paleoecology, sedimentology, geochemistry, and geophysics to reconstruct past environments and understand how marine organisms adapt to change to their environment. Particularly, he is focusing on ocean deoxygenation and how and why ecosystems recover after such changes or such conditions. His research interests include Cretaceous oceanic agnoxic events and Cretaceous mass extinction and modern and ancient foraminiferous geochemistry. Chris is currently working on several projects reconstructing the long-term changes in circulation and oxygenation in the Gulf of Mexico, the recovery of life following the end Cretaceous mass extinction, and changes in the solvent oxygen in the modern ocean. He graduated in geology from the University of Mary Washington, master and PhD from the University of Massachusetts, and currently he is a research associate at the University of Texas Institute for Geophysics. He participated in the Expedition 364 for the IODP. IODP is the International Ocean Discovery Program, and was the IODP lecturer for 2019-2020. Now, well, in this, he will talk about the foraminifera and how was the recovery of life after the impact crater. And in this context, I said a few words about the foraminifera in Spanish. Sorry, Chris. Los foraminíferos, ¿por qué son tan interesantes los foraminíferos? Regularmente cuando estamos hablando del impacto que tuvo el cráter, el asteroide hace 66 millones de años, nos referimos siempre a la extinción de los dinosaurios. Pero este impacto también impactó, valga la redundancia, a estos organismos, a organismos muy pequeñitos que viven en los mares. Los foraminíferos son organismos microscópicos del tamaño aproximado de arenas que aparecieron desde el Cámbrico y que desde entonces habitan en los océanos y que habitan tanto en los sedimentos como en la columna de agua. Por eso se les llaman formas bentónicas y planctónicas. Y se caracterizan por tener unos esqueletitos de carbonato de calcio. Estoy hablando más lento porque Chris me va siguiendo en el español. Tienen esqueletos de carbonato de calcio que les permiten que se queden en el registro fósil. Son ampliamente utilizados desde hace muchísimos años en la industria petrolera como marcadores de la edad de los estratos. Y también en las últimas décadas realmente son microorganismos muy taquilleros, si lo queremos decir así, utilizados en las reconstrucciones de paleoclimatología, paleoceanografía, paleoecología. Entonces, durante el impacto del Chicxulub no solamente se extienden estos grandes reptiles como los dinosaurios, sino estas pequeñas formas de vida que nos dan mucho de información sobre cómo fue el impacto en la vida durante ese tiempo y cómo, lo que va a explicar ahora Chris, es cómo se pudo haber recuperado la vida y las condiciones de este ecosistema marino. Yo agradezco a Chris realmente haya aceptado esta invitación en estas condiciones. Próximamente va a participar en otra expedición del IODP, entonces está por embarcarse próximamente. Thanks very much again, Chris, for accepting the invitation. I know that you are very busy with your new expedition and your new adventure. And please, let's move to your presentation. Thanks.
All right, thank you so much for that great introduction, Ligia. And thank you to you and Jaime and the entire Consorcio de Universidades por la Ciencia for inviting me for this talk. Also, thank you all for being here and for allowing me to give this talk in English. I've had the privilege of conducting my entire science career in English, which means that when it comes time to give a talk about science in Spanish, I'm hopelessly out of my depth. So thank you all for humoring me there. So let me just get some screen sharing going here. All right, I assume you can see that. So, yeah, the talk today is about the recovery of life after the end Cretaceous mass extinction. This is part of a talk that I've been giving for the past year as part of the Ocean Discovery Lecture Series that the U.S. Science Support Office for IODP puts on. And as part of that, they like me to talk about the history of IODP a little bit, which I'm very happy to do because I think it's really important. So I'm going to start off with just a little bit of background of what scientific ocean drilling does and why it's important. And then I'm going to talk a little bit about the background of the in-Cretaceous mass extinction, and then what we've learned from the new drilling in the Chicxulub crater off the Yucatan Peninsula. A lot of this work has come directly out of Expedition 364, and so I want to acknowledge some co-authors. Andy Frost, who actually wasn't on the expedition, but who worked closely with me on one of these papers we're going to talk about. Heather Jones and Tim Braylauer, who did the calcareous nanoplankton work from the expedition, and all of the Expedition 364 scientists. This is really a team effort. It's a large group of people that we worked with. It's pretty cool, actually. I'm a micropaleontologist, and there are people on this expedition that study impact craters on the moon. Really diverse group of scientists, and everybody you see in this picture contributed substantially to the science that came out of this expedition. So I would acknowledge everybody and their contributions to that. So scientific ocean drilling is something that's been going on for a while. Basically involves a drill ship. In this case, you see here the Joides Resolution. This is kind of the workhorse of scientific ocean drilling. It's got a derrick and a drill pipe goes down. It's not connected to the seafloor. The ship floats, and the drill pipe goes all the way down to the seafloor and drills into the sediments and the rocks, and it takes cores up the middle and returns them up to the deck. The current iteration of scientific ocean drilling, the International Ocean Discovery Program, has kind of four main research focuses. It uses to study the climate history of the Earth. Basically, the whole discipline of paleoceanography came out of scientific ocean drilling, and there's been a lot of contributions to that ever since about the climate and ice history of the Earth. The deep biosphere, there's actually microbial life that lives in the rocks deep within the Earth's crust, even kilometers below the surface of the oceans. And scientific ocean drilling has really been the way that we have learned about this and still study this today. Planetary dynamics, so volcanism and seafloor spreading and the creation of new oceanic crust, and then also geohazards, and particularly earthquakes and tsunamis that come from subduction zones as oceanic crust subducts below continents. These are all really fundamental Earth science things. Scientific ocean drilling is kind of our version of the Hubble Space Telescope or the Large Hadron Collider. This is how we do really big science, really fundamental research to answer questions and collect samples that we can never get otherwise, and that allow us to understand these kind of fundamental planetary processes that we would never be able to study without these cores from the deep sea. The ocean, of course, is 70% of the Earth's surface, and there is a lot that we still don't know about it. We've been doing this scientific ocean drilling, and I say we, obviously, I wasn't around in the 50s when this started, but all the way back in 1958, there was a project called Project Mohole, where they decided they wanted to try to drill to the Earth's mantle. And they thought, well, where's the easiest place to drill to the Earth's mantle? Where the crust is thinner in the ocean, so we should drill out there, which is a great idea, except nobody had ever drilled in the oceans before, except for platforms on shallow water. Nobody ever had a floating drilling platform that drilled into the oceanic crust. And so they had to invent all of these new technologies. How do you keep the ship on station? How do you even know that you're on station in an era before GPS? How do you trip string all the way down, drill string all the way down to the seafloor, and retrieve cores through kilometers of water? All these things. They didn't get to the Earth's mantle. They actually didn't even get close. I think they got like a couple hundred meters into the crust, and that was it. But they learned a lot about the composition of the seafloor and about the composition of the Earth's crust. Excuse me. And so they decided that it was actually worthwhile to start systematically drilling in the world's oceans and studying the seafloor. And this started as the Deep Sea Drilling Project in 1968, which was originally US funded and became internationally funded in 1973. It's been internationally funded ever since. And I'm not going to talk about all the different, contributions that scientific ocean drilling has made, but one of the very first drilling expeditions, leg 3 of the Deep Sea Drilling Project, proved the theory of seafloor spreading, which is a fundamental part of plate tectonics. And scientific ocean drilling has been making similar fundamental contributions to our understanding of the Earth system ever since. Sorry, you have to excuse me. I just went for a run earlier, and I've had something that's caught in my throat from some allergies ever since. Excuse me. So the Deep Sea Drilling Project was active until 1983. It was renewed as the Ocean Drilling Program in 1985, which was active until 2003. 2003, it was rebranded as the Integrated Ocean Drilling Program. And then in 2013 is the International Ocean Discovery Program. You might notice that we are running up against the end of this current iteration in 2023. And we are currently in the process of designing the next phase of scientific ocean drilling that should take us through 2050. This map is a little bit out of date. I think it's from 2018, but it shows up until that point, every single one of these dots is a site that has been drilled by scientific ocean drilling. So in one sense, a really impressive coverage of the whole Earth. But in another sense, there's a lot of white space on here. And these green and blue dots are some of the earlier expeditions that occurred back when technology wasn't as good and core recovery wasn't as good. And so these are also places we could return to and really do some more fundamental science. This is a map of active proposals that are in the system. So this is places that scientists have submitted really detailed drilling proposals to go and collect new data. And so there's a lot of proposal pressure. There's a lot of need still in the scientific community for continued scientific ocean drilling. There's a lot more to be done, basically, which is why we're working on a post-2023 science plan called a science framework this time. You can see the little logo here and the kind of new framing of life, dynamic Earth, climate, environment. It's been a long process of trying to figure out what we've learned from the last phases, what still needs to be done there, and what new questions have arisen from the science we've already done that require new research. The idea behind this is that if this is approved, if this goes forward, it would actually fund scientific ocean drilling through 2050. And so we could actually have some longer term drilling projects that actually require, decades to carry out, including perhaps drilling to the mantle again. We might actually get that done 100 years, or so after they started thinking about it. So anyway, that's just my very short spiel on scientific ocean drilling. I'm always kind of surprised how it's not as well known as some of the other big science programs that exist in other disciplines. I've always felt it's important to evangelize about it because it's really cool. Okay, so we're talking about mass extinctions today. Why do we care about mass extinctions? Well, first of all, they're cool. I think we can all acknowledge that it's just really interesting to learn about how the dinosaurs died and all that. But also, we might be living through a mass extinction right now. You might have heard the phrase the Sixth Extinction. So there are 5 major mass extinctions in Earth history. The end Cretaceous is the most recent. There's some feeling that we may or may not be currently living through a 6th extinction today. And the thing to remember about this is it hasn't happened yet. It is ongoing, but whether it actually turns into a 6th extinction or not is entirely based on our own actions over the coming decades. But certainly the way things are trending doesn't look good. So here's a plot that I thought was kind of surprising. This is terrestrial vertebrate biomass. So not insects or anything or plants, but just terrestrial vertebrates. At the start of the Holocene 10,000 years ago and today, this green is wildlife. So you can see, at the start of the Holocene, the quote unquote natural system looked like this. And then today, wild animals are such a tiny fraction of the overall terrestrial biomass. It's mostly people and domestic animals. Cattle, pigs, chickens, etc. Lots of other little factoids you can bring up here. 800 animal and plant extinctions have been observed since 1500, since the year 1500. Of course, those are only things that have been observed. You got to imagine that there's so many species even today that have not been described by science. And how many things have gone extinct without us even knowing that they existed? There are currently 17,000 threatened species, according to the International Union for the Conservation of Nature. Wild animal terrestrial vertebrate biomass has declined 60%. So we have, not necessarily extinctions, but the total populations of these groups has really dropped significantly. And modern extinction rates are between 10 and 10,000 times higher than they would be naturally, background extinction rates. What do we mean by that? Well, this is a plot of the last 66 million years, looking at the extinction of mollusks, which are these blue little shells, and marine vertebrates, so fish, basically. These little green fish you can see here. So this is the end Cretaceous mass extinction. This is at the genus level. So you have species and you have genera, which is one step higher up taxonomic order. So this is a little bit less detailed, but you can see 40 to 50% genus extinction at the end of the Cretaceous. And then over the course of the Cenozoic, the last 66 million years, relatively low extinction rates, what we call background extinction rates. There's a little blip here in the late Eocene. It's kind of big, but not huge. And then up through the modern, it's a couple percent of things go extinct in every one of these like 5 million year time bends. So natural background extinction rates are very low. You can make some forward projections, and you have to make certain assumptions to do this because we, A, haven't described all the species that exist in the world. B, even for the ones that we have described, most of them haven't been evaluated on if they're threatened or not, or their population's declining, or they stable, whatever. We just have these 17,000 that we know that we've evaluated enough that we can confidently say that they're threatened. So if you look at things that have been evaluated versus extrapolating to everything else, make some other assumptions about what the future might be. But the bottom line is the worst case scenario is in the future, the next hundreds to maybe 1,000 years, we could be right back at 40 to 50%, or I guess in this case about 40% genus level extinctions, which would put us right back at a in Cretaceous level extinction event, which again isn't certain. It's entirely dependent on our actions and our decisions over the coming decades, whether that happens or not. But the fact that it's a possibility is very concerning. And so obviously we're in the middle of this event. And if you want to understand how these things work, if you want to understand how extinctions occur, and equally importantly, how life recovers afterwards, you need to look to the past, to look to the past extinction events. And in particularly in this case, the most recent mass extinction is the in Cretaceous. So that's what we're going to talk about today. So the question here is basically, how and why does life recover after mass extinctions? And when we talk about recovery, we need to define that too, because it can mean a lot of different things. You can think about the recovery of habitability. So when was the environment able to support life again? So to use we're going to talk about the marine system, but there's a lot of really great evocative paleo art paintings of our mammal ancestors poking their heads out of their burrows after the extinction and the sky cleared, right? So here's this little mammal standing on a burnt log looking at this burned down forest. So you could say that this ecosystem has already returned to habitability in the sense that this guy can come up on the surface and he doesn't die, right? So that the short term, it's habitable. Organisms can live here. They have food to eat. They have everything that they need to survive. Another way to think about it is the return of ecosystem function. So how much energy is produced and consumed within an ecosystem? How many tiers of organisms of trophic levels are there in an ecosystem? So you have primary producers and things that eat them, and then things that eat those things, and things that eat those things, and so on up. Might not have the same total number of species, but all the same kind of functions are filled within an ecosystem as existed before. And then the longest term thing is the full recovery of biodiversity. So how long does it take to get back to roughly the same number of species that existed before the event? How long does it take to get back to that after the event? For the Cretaceous, particularly for foraminifera, the group that I study, it turns out it takes about 10 million years to get back to the roughly Cretaceous levels of diversity after the in Cretaceous mass extinction. Why is that is a great question. It's what we're going to kind of talk about today. What is the short-term, medium-term, and long-term recovery after the in Cretaceous mass extinction? What does that look like? So to talk about the Kpg boundary itself, Kpg's Cretaceous Paleogene. K for Cretaceous because we already have a Carboniferous and a Cambrian. So they were coming up with letter abbreviations for the geologic stages. They were running out of C's, and so they had to use a K for the Cretaceous. Anyway, this is a famous section in Gubbio Gorge in Italy, along a little road on a highway there. This is the Cretaceous, these rocks down here. This is the Paleogene up here. So dinosaurs, no dinosaurs, except these are marine rocks, so there's not really dinosaurs. So it's more like plesiosaurs and mosasaurs and ammonites, and all that stuff has gone up here. Of course, you'll notice looking at an outcrop like this, it's not like there's dinosaur bones sticking out of it, right? You can go, oh, look, there's a dinosaur. This must be the Cretaceous. Oh, look, there's no dinosaurs. It's not the Cretaceous anymore. So vertebrates, especially large vertebrates, are hard to find. And the benefit of looking at marine sections in particular is you can use fossilized plankton to get a much better idea of what's going on in these ecosystems. So these are planktic foraminifera. The scale bar down here is 0.1 millimeters. These are all about the size of a grain of sand, as Lihia very nicely explained earlier. And these are Cretaceous planktic forams. They were relatively large. They were very diverse. You can see all these cool shapes. These shapes all correspond to different life strategies, different niches that they occupied in the ecosystem. There are about 70 total species, and these things are found in these rocks. And for a long time, geologists have known that the dinosaurs disappeared around this level. Back to the early 50s and 60s, micropaleontologists have known that this was actually a really fast extinction event. There's this work that was done in this exact section here by Luderbacher and Promoli Silva back in the 60s, where they looked at these rocks and they found all the way up to this very last limestone bed right here. You find the full diversity of Paleocene or of Cretaceous planktic foraminifera. There's a little clay layer, and above the clay layer, these things all disappear. There's a few survivor species. They're all small. The scale bar is exactly the same size. They're very simple, and there are way fewer species in general than there were in the Cretaceous. A very rapid extinction event. So we've recognized for a long time that this is a very fast extinction event. But for a long time, people didn't know why. So back in the late 70s, early 80s, these two guys, Walter and Luis Alvarez, were working on this problem, and they're trying to figure out, well, why was this such a quick event? And they looked at this clay seam right here, and they thought maybe the clay layer indicated that there was a break in deposition. So the limestone that is made up of marine plankton just stopped being deposited for a while, and all that's left is a little bit of dust that drifts across the ocean and floats down to the seafloor, and you get this clay layer, and then limestone deposition begins again. So the idea was that there was a break in deposition, that there was some hiatus of a couple 100,000 years or something like that. And so they thought, well, how can we test this? And so they looked at an element called iridium, which is not found naturally in the Earth's crust, but it is found in space rocks. So asteroids, meteorites, things like that. Big ones, obviously, like the one that we now know caused the extinction event, but also every shooting star that burns up in the upper atmosphere, every little tiny grain of sand that disintegrates and then it hits our atmosphere has a little bit of iridium in it. And that little bit of iridium floats down and finds its way into the oceans and accumulates at a fixed rate very, very, very slowly. We measure it in parts per billion. And so what they expected to see was kind of background levels of iridium and then a little spike around the clay layer to suggest that 200,000 years were missing or something like that, and then back to background layers. What they found instead was a gigantic spike, about 80 times background, that you couldn't explain with millions of years of missing time, which didn't make any sense. They started looking at the clay layer more closely, and they found evidence of an impact event, other evidence of an impact event. Iridium obviously is a really strong one, but they also found shocked minerals. They found impact spherules. I'll show you some pictures of those in a second. They found other minerals that suggested that this was from an impact. And they thought, oh, wow, this must mean that this is what killed the dinosaurs. And they published this paper in 1980, and it was immediately a very controversial topic. And that could be a whole separate hour-long talk to just talk about the history of that. I won't get into that too much right now. But the idea that the asteroid has killed the dinosaurs has been around since the early 80s. This is a kind of a summary diagram showing just generally the changes that we see across the Kpg boundary. As a paleoceanographer, I love wiggles, and so I love showing these kinds of plots. These green lines are species of, whatever. They're not labeled as anything in particular, but just kind of showing. All the species we know from the Cretaceous that went extinct to the boundary, the Kpg boundary, almost all of them go right up to the boundary itself, and they all go extinct exactly at the same time. There's no stepwise extinction leading up to the main extinction event. Everything is fine, and then boom, really quick, there's a big extinction. A few species that were rare in the Cretaceous bloom in the earliest Paleocene right after the extinction event. They take advantage of the conditions and the lack of competition, and they bloom for a little while, and then they go back to being background species. Other species that were background species or generalists or whatever that survived start to diversify and lead to the diversity that we see throughout the Cenozoic after the extinction event. So these two things, the brief blooms of opportunists and the subsequent diversification, are kind of the things that we're really interested in here as we talk about the recovery of life after the extinction. This extinction event is exactly coincident with major changes in ocean chemistry. Carbon isotopes have a huge excursion. The weight percent of calcite in the oceans drops off really significantly, and this lines up exactly with a big spike in iridium. It does not line up very well with the pulses of volcanism, of volcanic eruptions in the Deccan province in India, which is the other hypothesis that a lot of people have have put out there, which is a reasonable one. A lot of the other major mass extinctions were definitely driven by large-scale volcanism. In this case, however, this is a 10-year-old paper, so we actually have much better dates for this onset of this quote-unquote main phase of decade volcanism. And it starts about 100,000, 150,000 years before the extinction event and continues several 100,000 years after the extinction event. And as I said before, there's no extinction that precedes this main event. It doesn't feel the earlier volcanism kind of turning on. It's a rapid extinction event and then a rapid recovery afterwards. How did this actually cause the extinction? Well, on land, it was a really bad time to be alive. The asteroid impact itself put a lot of debris up in the atmosphere. That debris all spread across the entire Earth and fell back to Earth, the larger pieces within a couple hours. There was so much of it that as it fell back to Earth, the friction of it falling heated up the atmosphere to the point that it was like being in an oven on broil. So hundreds of degrees air temperatures for at least a few hours. It's debated whether that was hot enough to cause forest fires over the entire Earth or over just North and South America. But either way, it killed everything that lived on the surface, which makes sense when we look at the things that lived on land that actually survived the extinction event. A lot of aquatic animals, turtles, freshwater fish, crocodiles, alligators, all totally fine. And if you lived in a body of water, you wouldn't have felt that thermal pulse. Our mammal ancestors that lived in burrows, totally fine. Birds that lived in caves and underneath rock hangings and things like that, also fine. Probably didn't affect the oceans at all, though. The oceans have a lot of thermal inertia, and turning up the air temperature on Earth for a couple hours really wouldn't affect them at all. But the other main impact of the impact, if you will, is along with all the heavier stuff that got thrown up in the atmosphere, there was a lot of dust, there's a lot of soot from the wildfires, and particularly bad luck for the dinosaurs, although good luck for us. The Yucatan Peninsula has a lot of evaporite rocks in the Cretaceous rocks that were deposited there before the impact. These evaporites have a lot of sulfur in them. The impact event vaporized the sulfur, put it up in the atmosphere as sulfate aerosols. And these are the things that when there's a big explosive volcanic eruption, cause the Earth to cool a little bit. And this put, hundreds of times more sulfate aerosols into the air than, say, Mount Pinatubo did. And it cooled the Earth. This is I love this climate model here. This is the Maastrichtian air temperature. So this is the red here, it might be hard to read, but that's 30 degrees Celsius, that's -60, the dark blue. This is the latest Cretaceous before the extinction event. This is the coldest year post-impact. And if you look at parts of North America, you're talking like -40, -50 degrees air temperature, which again, if you live in a body of water or a burrow underground, you can get by that for a few years. If you live on the surface, you absolutely can't. And the other thing that this darkness did, which lasted, maybe it wasn't total darkness, but it was at least very close to total darkness. For the darkest year post-impact year, the coldest year post-impact was 3 years after the impact event. So it didn't last very long, but it blocked out the sun, and that caused cooling, and it also caused the collapse of photosynthesis. So plants that use sunlight to make more plants and provide food for the rest of the food chain all collapsed, which caused the complete collapse of the food chain, which is probably the main cause of the extinction event in the oceans, if not also on land. So when the impact hypothesis was first proposed, one of the main criticisms was like, all right, smart guys, if it was an impact, where's the crater? They didn't know, obviously, it's a brand new theory. There weren't any known impact craters from the latest Cretaceous. There's a lot of worry that maybe the impact crater happened someplace that has been subducted since, or has been uplifted as a mountain range, or maybe it hit in the middle of the deepest part of the ocean and it didn't even form a crater. But as people started to look at these boundary sections, Kpg boundary sections, a lot of which come from scientific ocean drilling, as you can see from all these marine sections out here in the ocean, they started to notice a trend. At places we now know are very far from the Chicxulub crater, it's a very thin boundary sequence. So this is centimeters, so you got a couple millimeter thick clay, like we saw at Gubbio in Italy. This section is from Agost in Spain, but it's the same thing. Very millimeters thick clay with an iridium spike and some impact fallout just within a very small horizon. As you get closer to the crater in the western interior US, Gorgonilla Island in Colombia, there's a beautiful recently published section down there. You can see this is the impact horizon, and these are all impact spherules, classy beads, beautifully preserved. relatively thick. This is thicker now than what we saw in Italy. It's you know, centimeters, tens of centimeters thick, with a very clear layer of coarser grained impact debris, these larger spherules. We get even closer, like El Membral in northeastern Mexico, we see a meters thick deposit, spherules at the base, and then a high energy clastic unit that was deposited by a tsunami. And then above that is the clay layer of the atmospheric fallout that includes the iridium. As we get really close, like Gayal in southern Mexico, tens of meters thick, graded clastic unit that's ground flow from the impact event itself, and then impact debris, and then topped off by this clay layer, which is atmospheric fallout a few years after the boundary. So they started looking in the Caribbean and the Gulf of Mexico area in the 80s. Actually, two geologists published a paper about Penfield and Camargo, published a paper looking at this structure, which you can clearly see in gravity anomaly maps, magnetic anomaly maps, suggesting that it was an impact crater, but it didn't get a lot of attention at the time. And it got revisited in the early 90s. Pemex had drilled some wells in here because they saw a structure and they thought, oh, a structure, maybe there's oil in it. A reasonable assumption for most structures. But in this case, they were all dry holes. They put their core samples on a shelf and they moved on. And so the core samples were still sitting there for people to come back and sample and actually see impact melt rocks and impact tektites and shocked quartz and all these other impact indicators. And it was all dated to exactly the end of the Cretaceous. And they said, aha, there it is. There's the crater. This was published in 1991. And the first proposal to go drill the crater was in 1996. We got out there finally drilling offshore. The first proposal to drill the crater offshore, by the way, was in 1996 through IODP. Several holes were drilled on land. The Yaxcopiel-1 borehole, the most recent one in the early 2000s, which got some great stuff. But the idea of drilling offshore was that you have seismic data. We can actually see really clearly what the structure is. This is some of that seismic data. Our drill site is right there. This is the top of the impact sequence. This is the peak ring of the crater. Chicxulub crater is the only crater on Earth with an intact peak ring, which I could probably give a completely separate hour-long talk about all the cool stuff that's come out of that. Which unfortunately I can't. I would love to just talk about every single cool thing that's come out of this project because it's been really exciting. But anyway, I'll just leave that there. But we can see very clearly that what we're drilling into, and they actually were able to target a small depression on the top of the peak ring that was hypothesized to have the best potential to preserve the earliest post-impact sediments, which it turns out that it did, which is very exciting. So a lot of different objectives for this expedition, looking at the recovery of life afterwards, which is what I'm going to talk about, how do large impact craters actually form, and then how do they support microbial life, which are, I have colleagues working on those things, which has been really cool. I'm not going to have time to touch on it all today. Normally for these kind of things, we would use the Joydes resolution. Unfortunately, the water here was way too shallow. It was about 15 meters deep, which the draft of the ship is fine, but you have a drill strain coming out of the bottom. And if you're in 3 kilometers of water with a big metal pipe coming out of the boat and you move a little bit, it's not a big deal. The pipe can bend, it's flexible. But if you're in 15 meters of water and the boat moves a little bit, the pipe's just gonna shear. So the Joydes resolution wouldn't work. So instead we used the lift boat, in this case, this beautiful barge with feet called the lift boat Myrtle. And we just literally welded a drilling platform onto the front of it and used that to drill. Here's one of the feet. The water was so shallow, we were actually able to tie a GoPro camera to a rope and lowered it over the side, called it fish cam. Here's one of the fish. Here's one of the feet just sitting there on the seafloor. Took them a long time to get offshore and get set up correctly. And so they didn't have us leave the ship with them. And so they got out there, and then we left the port of Progreso on a crew boat. It took about an hour and a half to get off to the platform. And then they cranked us off on a little basket on the end of the crane. And we just kind of step on and you hang on, and they pick you up and they crane you around and put you down on the deck. Really cool way to get on board a ship. I've never done that before. Here's another image of the platform. Some people here to give you an idea of a sense of scale. So the first deck here was the galley where we ate our meals and hung out. Next 2 decks were sleeping quarters. They were 6-person bunks. Not, 6 people to a bunk, but 6 beds in a room. So, we work 12-hour shifts, and then you get off your shift, and then you have two other people in the room with you. Not the most ideal sleeping situation. There's also a common area outside that we could never use because there were always people sleeping around and all the bunks around. So if you want to do some quiet work, it was a nice place to sit, but it wasn't really a place to talk or watch movies or anything. Above that was the bridge. And then forward of that was a big open deck space that was covered in air-conditioned shipping containers that was our lab and science area. So here are these air-conditioned shipping containers. These are all, core curation, microbiology, sampling, micropaleontology, and so forth. When it was quiet outside, it was a nice place to catch a breeze, drink a cup of coffee. And then forward of that was the drill rig, which was, as I mentioned before, literally welded to the deck, the cantilever to drill floor off the front. This was a type of rig that's usually used in mining, to drill test holes where you're thinking about having a mine. And so it was perfectly suited to drill through hard rock, which was all the rock that we cored was very hard rock. And so we got amazing recovery. We got almost 100% recovery through the entire hole, which is really exciting. We got really good stuff. And then here's a drill floor looking straight down into the Gulf of Mexico and into the heart of the crater. Cores would come on deck. We got 3-meter cores. We'd split them, and then we would sample them, and then we would do some preliminary work on the ship. Here's me looking at a microscope, looking at microfossils. So what did we find? Here's the borehole again. So we didn't take a core for the upper 500 meters just because drilling is really expensive and takes a lot of time, and we wanted to use our time wisely and drill more into the deeper part of the crater. And so we didn't take any cores up here. We just blasted through it really quick. Started coring about 500 meters below the surface. Got 115 meters of post-impact sediments from the Eocene and the Paleocene, including a nice record of the Paleocene-Eocene thermal maximum. My colleague Van Smith just published a nice paper on that in Climate of the Past. And then we got 130 meters of impact rocks, impact melt rock and melt-bearing impact breccia called suovite. And then below that, we found uplifted basement granite that actually comes from like the middle to the bottom part of the crust, uplifted kilometers by the impact event. And it's complete, it's really fractured. It's cut through with lots of melt dikes, really cool stuff. I just want to show some pictures because it's really cool. This is some of the intact granite, some more fractured granite with a big melt dike cutting through it. This is part of the melt sheet, some of it which is just black and massive like this, other parts of which have some texture to it, nice schlieren through here. And then this is the base of the breccia, the suovite melt-bearing breccia that forms the base of that unit. Really coarse cobbles at the bottom, and it gets finer as you get further up section. This is an old kind of preliminary figure, but I think it's a really nice one that summarizes this, because you can nicely see as you go up section, you go from cobbles size grains to small gravel to sand size grains, kind of basically moving up section. This is a plot of the largest grain in each section of core, and you can see kind of these general trends. This is all ground flow, and then there's an erosional contact right here, and then the rest of this is deposited by the tsunami. So you have ground flow coming back in the crater, and then about an hour after the impact event, there's a great paper by Sean Gulick and others for the whole science party. It was published last year in Proceedings of the National Academy of Sciences. It very nicely steps through this whole sequence. It's awesome. I definitely encourage you to check that paper out. But the tsunami comes back in, and you get some submarine deposition there. Above this, at the very top of this fine-grained breccia unit, as you get the finest grained stuff, there's this you can't see, but this is cross-bedded from another tsunami wave coming through. And then above that, you get this fine-grained clay stone, this brown clay stone that we call the transitional unit. And this is basically from settling. So after the tsunami and all the Seiche waves and everything else kind of ended, you had a bunch of just muddy, turbid water in the crater. And this muddy, turbid water would have slowly settled out of suspension. And that's what this unit is. And at the top of this unit, we find the iridium layer. This is the spike of iridium. This is from a presentation of the Lunar Planetary Science Conference by Stephen Godaris and others from last year. And this is the enriched iridium layer. And this is really exciting because we know from models that the iridium layer, that the cloud of dust that spread across the world and settled back out of the atmosphere would have been up there for a matter of years at the most. I think the outside estimate is like 10, but more realistically like 2 or 3, which means that this whole settling layer, this whole transitional unit was deposited within a few years of the impact. And the reason that's so exciting is we find evidence for life in the crater within that interval. So within a few years of the impact, while the sky is still dark and the extinction is still going on, there's life colonizing the crater itself, which is just really exciting and really cool. So the lower part of this, there's nothing. The first weeks, months, maybe year, it's really hard to figure out a age model for this part of the unit because it's so far beyond any sort of geologic timescale or tool that we can use to figure out the relative rate of deposition. We can say this whole thing was deposited in less than a few years. We know that this stuff was deposited within hours. And so there probably is a transition in here where maybe this bit was deposited in days to weeks to months, and then this is in the year. Hard to say for sure. But anyway, the lower part has no evidence, direct evidence for a lot of life. There actually is a little bit, I'll talk about that in just a second. But up here towards the top, we start to see burrows. You can see these here. They're not the prettiest burrows. When I give this talk in a room, they never look that good on a projected screen. Hopefully on your computer screen, you can see these little white-brown smudges in the middle of these circles. But they're definitely burrows. And the great thing about burrows is they can't be reworked from the Cretaceous. When we look at survivor species, it's kind of tricky because they survived from the Cretaceous, which means you find them in the Cretaceous and you find them in the Paleocene. You can never be 100% sure that the individual you're seeing weren't reworked from the Paleocene. We know that burrows can't be reworked because they're primary sedimentary features. We also know they didn't come down from above, first of all, because these are horizontal burrows and not vertical burrows, so they wouldn't have come down tens of centimeters. But also as we get closer towards the top, right below this contact, we actually see some that did come down from above, and those are all infilled with the same material that occurs above. So the fact that these are infilled with material from the same layer indicates that these are contemporaneous. These were formed at the same time as this layer was deposited, which means there was life in the crater at this time. We also see this from microfossils. This is a plot of foraminifera per gram of sediment, total foraminifera. So these are mostly reworked things from the Cretaceous, probably. At least down here, they certainly are. There's actually 3 data points down here that are missing from this plot because they all have thousands of grams, or individuals per gram of sediment. And these are all Cretaceous species that we know went extinct at the end of the Cretaceous. So they're all reworked. That drops off really strongly right about here as the energy drops off, which means there's less energy, less possibility of reworking. And also at this level, as things start to drop off, the percentage, this is a plot of the percentage of the total foraminifera population that is composed of survivor species, which, as I mentioned earlier, you can never be 100% sure that they weren't reworked. But because they start to occur in some abundance at this level, once the total reworked assemblage has declined, then you start to see this increase in abundance, which suggests that these are actually real survivors that are living in the crater at the time. In particular, this orange line, which is not super abundant at all through this whole interval, but is increasing in abundance up section. This is a genus called Wimbleditria. And in the Cretaceous, these lived in marginal marine environments. You find them in inner shelves, in continental seaways, things like that. You don't find them in the open ocean. And at the end of the Cretaceous, the Chicxulub area was the open ocean. The closest land was probably 500 kilometers away. So you would not expect to see any Wimbleditria. You wouldn't expect to have any Wimbleditria close enough to even be reworked into the crater. Excuse me. Which means that these are real survivors that are coming in. We see this also with calcareous nanoplankton. This is a genus called Micula, really, really small. It's a survivor species, and it increases up section within the transitional unit. Calcareous nanoplankton, of course, so we have foraminifera are zooplankton. They eat other plankton about the size of a grain of sand. Calcareous nanoplankton are phytoplankton. They're primary producers. They're basically plants. And so here's a foram that's the size of a grain of sand. If we zoom all the way in on one of these pores, there's a calcareous nanofossil right there. So way smaller. Can't see them with your naked eye. Although, as everybody likes to point out, when they bloom, you can see them from space. Here's a bloom of Meliana huxleyi in the modern English Channel. So we see a lot of survivor species in here. And again, you wonder, well, are these reworked from, just latest Cretaceous things that are reworked into the crater? And so to figure this out, Tim Braylauer did this, I think, really cool plot where he looked at the red squares are Chicxulub, abundances of these species found in the Chicxulub crater, and the blue squares are abundances of these species that are found in lots of different latest Cretaceous sites. And so if what we see in the Chicxulub crater is reworked from the latest Cretaceous, we'd expect these squares to plot firmly within the rest of the latest Cretaceous sites, right? That's what a reworked assemblage would look like. But instead, we see anomalously high abundances of some survivor species, Retacapsa, Watsonaria. We see anomalously low abundances of species that went extinct in the Cretaceous. Prediscosphaera, Ipholithus, Chiastozygus, Archangel Sciella, again, which indicates that these are real in situ assemblages, maybe with some minor component of reworking is evidenced by these things in a low abundance, but most of the assemblage is dominated by survivors that are living within the crater. We also recently have found evidence for cyanobacteria and other bacteria living in the crater in the transitional unit as well. My colleague Bettina Schaefer and others just published a paper in Geology this year looking at biomarkers. They found evidence for cyanobacteria. Total bacterial activity is high in the transitional unit. Evidence for certain kinds of cyanobacteria in the early part of the transitional unit as well. So again, a biomarker indicating these things bloomed in the crater, even at the period of time when the sky was still, darkened with soot and sulfate aerosols, which means that the sun wasn't blocked out entirely. If you have photosynthesizers that are managing to get by in the crater and elsewhere in the ocean, surely as well. Maybe sunlight was to decrease 90%, but it wasn't totally wiped out. We also see other evidence. This is more work that Tim has done, and this is in revision right now for a journal called AGU Advances, where he actually found evidence for some of these survivor species looking in more detail since our first paper was published, right at the base in this interval that's hours to days after the impact, and you still see these things living in the crater, which is really exciting. And then higher up, above the iridium layer, we start to see a lot of diversity of new planktic species, foram species coming in within thousands of years of the impact. A low diversity of calcareous nanoplankton, and actually evidence fish bones, crustacean appendages, showing again a relatively high diversity of life in the crater. The place that basically you would expect the environmental effects of the impact to be the worst was habitable very, very fast. Which tells us in general that the oceans were habitable basically right away. Like the extinction happened really quickly, things went extinct, and then within a few years, the oceans were habitable, life could survive. So the environmental changes caused by the impact were very, very short. The ecological changes lasted much longer. We can think about ecosystem function, in this case, in terms of export production in the oceans. There was a decline in export productivity that lasted about 1.8 million years. So here's just a kind of a summary figure. This is from a nice paper by Zhang et al. In 2018 that summarized, that reviewed basically our knowledge of the biological pump. But you have phytoplankton in the oceans that take up CO2 and sunlight. They die. Some of them get eaten in the upper oceans. Some of them sink down and get eaten in the lower oceans, or get remineralized, or turn into dissolved organic carbon. They never quite make it to the seafloor. A small percentage of that makes it all the way down to the seafloor and gets preserved. In the modern ocean, we can just observe this process, but we also know that there's a carbon isotope gradient between the surface and the deep sea, and we can use that carbon isotope gradient that's caused by this biological pump to reconstruct the biological pump in the past. So carbon isotopes, right? There's carbon-12 and carbon-13. There's carbon-14, which people use for dating. It's not stable. It doesn't last very long. But 12 and 13 are both very stable. Carbon-12 is preferentially used by organisms undergoing photosynthesis. It's a little bit easier to take in carbon-12. Requires a little bit less energy because it's slightly less heavy, one less neutron. And so it's a little bit easier to incorporate that into your cell if you're a photosynthesizer. And so plants photosynthetic plankton are really enriched in carbon-12 and depleted in carbon-13. Those plants die, some percentage of them sink all the way to the seafloor, which removes carbon-12 from the surface ocean and takes it to the deep sea, which leaves surface waters relatively enriched in carbon-13 and depleted in carbon-12. We can measure this from foraminiferal calcite, which incorporates carbon into the shell of the foraminifera. We can look at planktiform, which lived up in the surface waters. We look at benthiform, which lived on the seafloor. And we can see in the modern ocean, it's about a 2 per mil gradient from the surface ocean to the deep sea. At the end of the Cretaceous, this is from a really cool paper by Heather Birch and others from 2016. At the end of the Cretaceous, this gradient existed. Part of the strength of the gradient, the whole 2 per mil, part of that is because some of these plankton actually have photosynthesizers in them as well that actually cause them to be slightly even more depleted. So if you look at photosynthesizing latest Cretaceous plankton, it's again about a 2 per mil difference. If you look at non-photosynthesizing latest Cretaceous plankton, it's about a 1 per mil difference. The photosynthesizing things went extinct at the Cretaceous, but not everything did. And we can see from the things that survived that the gradient collapsed right at the boundary and stayed low and began to recover very slowly. And it took about 1.8 million years to get back to what it was before the end of the Cretaceous. And then as that gradient continues to increase, this is actually evidence of the reappearance of photosymbiosis in plankton foraminifera, and then eventually you get back to this higher, roughly 2 per mil gradient that existed before. But what this tells us is that the biological pump was basically recovered by about 1.8 million years after the boundary, which again, we're not super fast, but not, as we mentioned before, that recovery of life took 10 million years. Recovery of diversity, rather, took 10 million years. So the ocean's ecosystem function was back to normal relatively quickly as well, which is why it's so surprising that diversity took about 10 million years to get back to what it was before. This is a plot from a paper we just published in Annual Reviews of Earth and Planetary Science, working with Paul Bound, who provided this nanofossil data, my colleague Andy Frost, who had this great planktiform diversity data set that he recently updated. We used in here as well, and also with Shelley Hull at Yale University. And so we all worked on this stuff together, and so this is the record of calcareous nanoplankton. Calcareous nanoplankton anyway, evolved in the Triassic, increased their diversity throughout the Cretaceous, experienced a huge extinction event at the Kpg boundary. I should mention, by the way, this is species level diversity. So if you remember back to that plot that I showed on the page with the Sixth Extinction book, and we looked at the little fish and the little shells, that was genus level diversity at about a 5 million year time step. So this is species level diversity, so it's taxonomically much more precise. And these are million year size time bins. So we can see a much more detailed change in diversity through time as well. And this is really exciting because plankton are probably some of the best preserved, have some of the highest resolution taxonomically and temporally highest resolution records of any fossil group. And so we can see in really high detail the changes they went through. We can compare these to different events that have occurred in the deep past and understand how, say, oceanic anoxia or acidification or other things affect their diversity. That's what the point of the rest of this paper is, which I won't get into, but this is the Triassic-Jurassic Boundary, the Toarcian oceanic anoxic event. These are Cretaceous oceanic anoxic events. Here's the Kpg boundary, Paleocene-Eocene thermal maximum, the Eocene-Oligocene boundary, which is also a big extinction for the plankton. Anyway, and the other thing about even like a million year time bin is really, really good for paleodiversity studies. But even then, we actually lose some resolution here because the extinction was so fast and the recovery was so fast. You actually had, for planktiforms, for example, 90% of the species went extinct. You had 12 new species that came in within the first couple 100,000 years. So even within a 1 million year bin, we lose that full drop in diversity. If you actually had the full extinction, it would actually come all the way down to about here. Same thing with the nanoplankton. So don't think that they actually didn't go as extinct. They actually both went all the way down. But anyway, nanoplankton, plankton foraminifera also increased through the Cretaceous. Both of them suffer a major extinction at the Kpg boundary. Both of them have low diversity for a while. Forams, by 10 million years later, get back to middle Cretaceous levels of diversity. Nanoplankton have a bit quicker, initial rebound, but even for them, it's about 10 million years to get back to that latest Cretaceous level of diversity. So there's the Kpg boundary, and you've got, again, this really diverse, complex multi-layered ecosystem, and then the survivor species are these low diversity, non-complex things that are much smaller. We see this in other groups as well. This is another from an older paper, lower resolution data, again with the calcareous nanoplankton. Dinoflagellates also had a similar drop that also lasted about 10 million years. Lower resolution, but it's still, there's 2 data points in there. This isn't an artifact, just an artifact of sample resolution. Diatoms had just started to evolve and started to really enter the fossil record, and it's unclear to me if this bump is an artifact of this is where everybody looks for fossils because it's the Kpg, so there are more diatoms that are known, or if they actually had a little bit of an increase right there. But for most groups, it's about a 10 million year delay. And in fact, a 10 million year delay in the recovery of diversity is something that is seen after every major mass extinction event of the Phanerozoic, the past 500 million years. This is a great paper by James Kirshner and Ann Weil where they looked at this. This is family diversity and genus diversity all the way back to 500 million years ago. So these are the 5 major mass extinction events. And this is the percent extinctions. And then this plot is the percent originations, so the new species coming in. For each of these, there's a gap of about 10 million years. Peak in extinctions, peak in originations, peak in extinctions, peak in originations, peak in extinctions, peak in originations, so on and so forth. They did some math and lined all those things up, and again, it's about a 10 million year delay for each of these major events. Well, why is this? This Kirshner-Weil paper offered an explanation for this as well, which is that basically that ecological interactions are complex, ecosystems are complex. Ecosystems are the reason we have this, or complex ecosystems are the reason we have high diversity, and so it takes a long time to rebuild that complexity. You can think about extinctions eliminating ecospace, which a lot of times is, especially in the past, we're talked about as niche space, an ecological niche that the species occupies. We tend to think of an ecological niche like a parking space, right? That you can park a species in, and that species goes extinct, and now that parking space is open, and another species can pull right in and fill it up. But that's not really how it works. When a species goes extinct, it takes its ecological niche with it. And so an easier way to conceptualize this, I say easier, fully recognizing this weird, complex figure that's sitting on my slide right here, is to consider this as ecospace. So a multidimensional space that includes all the different things within an ecosystem and how they're related to each other. I spent a long time when I was putting this talk together trying to find a good visual representation of ecospace, and this is the best I could do, which I'm probably a failure on my part. I probably could have kept looking even longer, but I do like this because it nicely shows how complex it is, at least. But I'll walk you through this. So each of these dots is a species, right? And so this is a multidimensional space. You can think about the things closer to the middle as being more simple, things further out being more complex, or filling up more discrete, more sophisticated or unique parts of ecospace, right? Each of these lines is how these different species are related to each other. Now imagine that 75% of these dots disappear, particularly the ones around the outside. They're the most complex, have the most specialized habitats, right? Lifestyles. So then you have a smaller cluster of dots in the middle. It's going to take a long time to rebuild that complexity, to rebuild those dots, to kind of get this kind of whole ecosystem back together. And again, this is a very complex figure. So what I want to do is kind of step through maybe a more accessible example. At least it helps me think about this a little bit better. So imagine a coral reef, right? One of the most biologically diverse places on Earth. Thousands of species, tens of thousands of species living in one ecosystem. And it's built entirely on these interactions, right? You have dozens of species of coral that actually literally form the reef itself. You have different species of coral that are adapted to different parts of the reef. Certain species of coral that live on the forereef, that live on top of the reef, live in different parts of the lagoon. Certain species of coral that encrust other certain species of coral. On top of that, you have a huge diversity of invertebrates and vertebrates that are adapted to living in particular parts of the reef, that eat particular corals, that eat things that eat particular corals, or hide an anemone that lives on a particular coral, right? These are all very multi-layered, complex relationships. You have larger predators that live off the reef as well. And these larger predators, like the shark, are basically their own mini ecosystems themselves. Here's a shark, and the shark has parasites, and it's got little fish that eat parasites off the shark. So layers upon layers that build up this huge, amazing biodiversity we find on reefs. Now, imagine that all the reefs on Earth go extinct because of, I don't know, let's just say the oceans get too hot and they all bleach, to pick a random example. And it's not like a modern bleaching event that we see right now, where like one reef over here bleaches and the other one is fine, and it can kind of re-diversify back, disperse, coral polyps and regrow the same spot. The species are all gone all over the world. So there's just a few species of coral left. There's a few species of fish and invertebrates that live in the coral as well that are left. It's going to take a long time to rebuild the full complexity of the reef ecosystem, because you've got to basically rebuild all those levels, and it takes a while to get there. You got one species of coral that's got to basically diversify to refill all those different parts of the reef that its relations had before. Then you've got to have things that eat those corals start to evolve, and then things that eat the corals start to evolve, and so on and so forth. So the idea that Kirchner and Weil proposed in their paper was that 10 million year delay is just caused by how complex ecosystems are and how long they take to rebuild. This is one of those papers that you read and it's like a light bulb goes off in your head and you go, of course, that makes perfect sense. So I was working on this problem. I read this paper a few years ago, right after the drilling expedition, aware that there was a big delay in the recovery of diversity of plankton. And I was really excited about this paper. I did the thing where you go to Google Scholar and you click on the papers that cited this paper link, because I was like, it's 2000. I'm sure we've learned so much since then. And the paper has been cited, but nobody had actually followed up and actually tested the idea that they were proposing. And so at AGU a few years ago, I was having brunch with my friend Andy Frost, telling him about all this and lamenting that nobody actually tested it. And Andy goes, we can test that. And I was like, oh, really? Tell me more. So Andy had a data set from his master's program where he had measured every single planktiform species that existed. So he had a bunch of different measurements. He had all these morphological measurements of forams. Planktiforms are relatively simple organisms, right? It's one cell, they have the shell, and that's it. And so the idea is that you can use the shape of the foram, the complexity of the foram shell, as a proxy for its habitat, right? So based on the assumption, which is a pretty key assumption with this whole thing, but based on the assumption that they don't form these shapes for fun, for just randomly, that these shapes have some sort of purpose, which kind of makes sense because a lot of these shapes repeat through time after extinction events. But that these shapes actually form some sort of purpose, that they're related to their lifestyle, then the variety of shapes that occur within the overall population of plankton, the number of species, tells you something about the size of the ecospace that the plankton occupy. Basically what we're doing here is using morphospace as a proxy for ecospace, right? And this makes sense qualitatively. If you just look at latest Cretaceous, here's a few example latest Cretaceous planktiforms. They're big, they're weird, they have a lot of different shapes and bumps on them and spines and shapes of their aperture and a bunch of other stuff. The things that survive the extinction are all very simple. The things that evolved from the things that survived the extinction are also very simple, slightly more complex, but not that much. And it's not until about 10 million years later that you start to get these more complex things coming back in. Looks great qualitatively. So we wanted to look at this quantitatively. So this is Andy's data that he had measured. You don't need to worry about this in particular, but the nice thing about forams is because they're so simple, it's really easy to come up with a data set that is not easy. I think Andy'd be mad at me if I hear me say that it was easy because he did a lot of work when he's doing this originally. But it's possible, it's relatively straightforward to come up with a data set of different measurements of forams you can compare to all the other different species. Measure the shape of the shell, the size of the aperture, angularity of the views, and all this other different stuff. So this plot is, this is test complexity. So each of these gray lines is a species. The length of the line segment on the x-axis is the length of time that it lived in the world, right? So this is time on the x-axis, and there's complexity on the y-axis. So it's position up here, more complex up towards the top, less complex towards the bottom. We can look at mean and median complexity, and we can see a huge drop off in both at the end of the Cretaceous, and it comes back up and peaks again at the end, like 10 million years later at the end of the Paleocene. Looks good, doesn't actually test the hypothesis though. To test the hypothesis, what we needed to do was look at this factor of test complexity over diversity. And the idea here is that if ecospace is being filled linearly, so every time you add a new species, ecospace expands just a little bit. Every new species is a little step, and it goes a little bit further each time. You would expect to see basically a flat line, right? As you have that, as those two terms change together, which is what we see in the end of the Cretaceous. This is the highest diversity of planktophorians that has ever existed, and they're the most complex that they've ever been. But you can see this line of complexity over diversity is basically flat. If, on the other hand, complexity is leading diversity, so there's an evolutionary innovation that opens up a bunch of new ecospace, and then species come back and fill that in. So complexity jumps, and then species fill in behind. Basically, the complexity, the ecospace occupation, so complexity being a proxy, basically being a way to say morphospace occupation, which is a proxy for ecospace occupation, is the thing that's holding it back. We'd expect to see these really high values as it jumps up, which is exactly what we see through the Paleocene. Overall, there's a big spike at the start as you start to get the recovery, but over the whole course of that 10 million years, the mean complexity over diversity is relatively elevated, and it goes back to normal about 10 million years later, which means after about 10 million years, ecospace expansion rate wasn't the thing that's holding back diversity anymore. And we can look at this with other groups as well, but I've been talking for about an hour, so I want to wrap it up. But what this means is, the key takeaway point here is that these pulses of evolutionary innovation are the things that open up new ecospace, and then it's filled out behind. What this means is that there's basically a fundamental speed limit on diversification, right, after a mass extinction event. The Kirshner-Weil hypothesis is correct, at least within plankton foraminifera, the thing that we could test here needs to be tested for other groups. If anybody works in other groups is listening, it would be awesome if this was tested for those things as well. But what this means is there's a fundamental speed limit to diversification. It takes about 10 million years after an extinction event to get back to pre-extinction levels of diversity. So to wrap things up, there's a long delay in the recovery of diversity after all extinction events, including the Kpg. Delay is not caused by environmental forcing. The oceans were habitable, including ground zero in the impact crater within years of the impact. And the ecosystem function had returned within 1.8 million years after the impact. And rather, it was internal macroevolutionary processes, specifically the need to rebuild ecospace, that kind of dictates the speed limit after extinction events, which means obviously species that go extinct today are gone forever, but biodiversity that's lost today is going to take a long time to be replaced. If we actually wind up living through a sixth extinction of Cretaceous proportions, the decisions that we make now are going to affect the biosphere for 10 million years in the future, which is way longer than climate change. The bomb spike and CO2 and all that other stuff will be gone. The ice sheets will be back well before the biosphere recovers, which is a sobering thought. But again, the thing I want to leave you with is that it hasn't happened yet. And it's our decisions over the next decades that will determine whether it does or not. So with that, thank you very much for your attention, and I'm happy to answer any questions.
Many thanks for that wonderful talk. I think it was really nice to hear about both the logistics as well as the actual effects of meteorite impact and this more wider perspective of events after a mass extinction. So I'm actually quite intrigued by this magic number of 10 million years that it takes to recover biodiversity after a mass extinction. And I'm a bit puzzled by it because every single mass extinction we've seen has been associated with different causes. And also there is a huge range in the percentage of species lost in each case. So is this 10 million year more like a ballpark figure, or do you see that it's pretty constant? Yeah,
so the Kirshner-Weil, when they did the math for all these events, it was, in 10 million years is like the average, it's like 8 to 12 or something like that. But the interesting thing to me, and I think what makes the in Cretaceous such a good test case for this, is these other extinction events that were caused by longer term environmental changes like massive volcanism or huge glaciations or whatever were slower, and the environmental recovery after was slower. And so it's easy to make the argument in those cases that it's not just macroevolutionary processes that's holding things back. It's you know, the recovery of the environment after, say, the Siberian traps finally stopped erupting after the Permian mass extinction. With the KPG, and I mean, that's why I think it's really important to talk about, you know, the quick recovery of life and the habitability of even the crater right after the impact. That the effect on the environment after the Cretaceous mass extinction was not very long at all. It was really, really quick. It was, you know, within years, even the crater was habitable. Within millions of years, ecosystem function had returned to normal. Those are all the sorts of things you could imagine holding back diversification. And so if none of those other things are actually holding back diversification, it becomes a question of, well, why does it take so long for diversity to actually get back to where it was before? And that's where you start to think about these internal evolutionary processes. So, I mean, that's a really good question to compare it to other extinction events that had these longer term things. But even in those cases, it's not like it took 15 million years or 20 million years after the Permian. Like, it's still the peak in originations and the peak in extinctions are always spread out by about 10 million years.
Just that I am muting my microphone as much as I can. So we have another question asking about the recovery of life in continental land. So you were telling us about the effects of the extinction event and then the recovery based on samples presumably from oceanic environments, but what happens in land?
Yeah, basically similar processes occur on land. Actually, there's a quicker recovery of photosynthesis and primary production. There wasn't a big extinction of plants, which is maybe surprising when you think about like global wildfires and all that stuff. But forest ecosystems, plant ecosystems are kind of used to dealing with fire. And so they have seeds that can survive. And so even if all the trees died, the seeds are still there. And actually what we see pretty cool on these records of pollen that we find in nearshore environments right after is what we call the fern spike, which means, and if you remember that picture of the little mammal standing on the burnt log, there's a bunch of ferns on the ground. Basically for the first hundreds or maybe 1,000 years after the boundary, there's just ferns everywhere. It's like the first thing to come back really quickly. So there was food for things to eat really right away. And then you start to see the recovery of trees, forests coming back relatively quickly as well. So, you know, in the oceans, the problem was, at least the fossils that we can see, and this is the other thing, is we can only look at fossils that have hard parts. We have some biomarkers for cyanobacteria and stuff, but we mostly look at fossilizing plankton. We know the fossilizing plankton had a big extinction, so it took a long time for the primary producers to come back. But on land, you know, forests and grasslands, it all came back really quickly. Diversification of mammals kind of happened apace. I think it's been a little while since I read this paper, but I think it is like, I think that mammal diversity starts to peak, and you can't make that direct comparison because they're not dinosaurs. So the mammal diversity before the extinction was not the same, but mammal diversity starts to peak around the PETM, and it's not until like the late Paleocene that it really starts to take off. And you get I believe that's also when you start to see the evolution of primates. It's also pretty cool, but yeah.
Yeah, thank you very much. I think it's quite interesting to hear about this fern spike. A few weeks ago, we had a presentation by Dr. Daniel Field, and he and colleagues were we're looking at that fern spike and the effects on the evolution of birds. So I think he might still be watching us. He was watching your talk earlier, and so hi to Daniel. Yeah. And so what he was saying is that as all the trees died, only the non-arboreal birds were able to survive. But I think what you're saying is quite important, that even if all the trees died, there was a kind of seed bank that remained on the ground. So yeah, we have a question from Mario Rebolledo. So Mario, hi. It's really great that you're here. So he is saying that it's a great message about the biodiversity recovery across time. And what he's saying is, what are your thoughts about this paper? I didn't read the paper, but I did saw a note in the newspaper about how the Deccan traps and the eruptions helped in the recovery of life. I think people looking at them have long been trying to establish a link with extinctions. So I guess now they are switching into trying to establish a link with radiations.
Yeah, this was a cool paper that I saw just got published recently, that they looked at they looked at ecosystem modeling for dinosaurs, and they basically looked at you know, if you, based on where dinosaurs lived and what the kind of presumed environment was like, how much could you change the environment and have the dinosaurs still be able to live there? And so they found with the cooling that occurred after the impact, that really shrunk the dinosaurs' environment quite a bit, and the environment of everything else that was living as well. And the continued eruption of the Deccan traps and all the CO2 that put in the atmosphere actually counteracted the cooling and warmed the Earth enough that it didn't cause a worse extinction of, you know, like avian dinosaurs, like birds, or other terrestrial animals as well. So it helped help reduce the severity and the length of the cooling that occurred afterwards, and so kind of help recovery along, which is pretty cool, I think. There's been a lot of really good Kpg boundary papers that have come out in the last year, looking at ocean acidification, looking at temperature change, and all of them show almost no change leading up to the event. So even when Deccan volcanism has started and is going really strong, there's no change. There's a big change right at the boundary, and then a really quick rebound, and then back to normal as Deccan volcanism continues along. And so putting that massive volcanism that was occurring through this whole time in context of a recovery of life is really cool, I think. It is. I think it's really interesting to kind of have that switch from like, oh, it drove the extinction, to actually probably helped the extinction be less severe.
Well, I guess they were defeated on the first part. So we have a question from Nadia Maturano Ramirez, and she's asking that if you are trying to equate the impact of human activity to the impact of a giant meteorite hitting the Earth.
Yeah, actually, sort of. So the thing is, when you're finding analogs for what's going on today with human activity, there is no perfect analog. There's no perfect analog for the type of climate change that we're causing. There's no perfect analog for the type of extinction that we're causing. So we can look at the geologic past for partial analogs. And so, for example, with climate change, a lot of people like to look at the Paleocene-Eocene thermal maximum, which was a rapid warming event that was caused by a rapid release of CO2. That rapid release of CO2 occurred on the scale of thousands of years, which is still, you know, orders of magnitude slower than what we're doing today. But it's the fastest event of its type in the geologic record. The cool thing about the Cretaceous mass extinction is it's actually, at least of major geologic events, it's the only thing that's happened faster than anthropogenic global warming has. And so it's a really good way to understand how really fast ecosystem changes can cause extinction. And when you have a really quick event, which is, I mean, geologically speaking, what we're doing to the planet today is going to be relatively quick. If we stopped emitting CO2 right now, I think it would take a couple thousand years for atmospheric CO2 to kind of go back to normal, and tens of thousands of years for ice sheets to go back to normal. I mean, which on human timescales is a long time, but on geologic timescales is really, really fast. And so it's not exactly the same, but it's a good way to try to figure out at similar timescales, how do ecosystems respond and how does recovery happen? So it's a good, yeah, I wouldn't say that I'm directly comparing it, but it is probably the most useful analog extinction-wise in the past.
Okay, so I think that's a warning for all of us, and to really take stock and think about what we're doing. Patty Cerrillo is sends her congratulations, and she's saying that she understood from your talk that this anthropogenic era is leading to a new mass extinction, and she's asking whether this splurge of new species is already happening, or whether we are just in a phase of getting the existing species to become extinct, and only then we'll go into radiation mode.
Right, that's actually a really cool question, because I think that stuff is fun to think about. I really like to think about how species evolve and how they go extinct and how they change. Forams are really cool for that. There's actually great records, longer timescales, but on thousands of years timescale, where you can see a species change, where like it's, it basically changes from going really skinny to really short or something like that. And you can actually observe the change in a core, looking at a different population. But this might probably be a better question for a modern ecologist, but my assumption would be, yes, there is. I think that there's always evolution happening, slowly at background rates, probably faster now because we're causing pressures on a lot of organisms, and a lot of things are going to go extinct because they can't keep up with those pressures. But other things that manage to adapt are going to very quickly become new species, and very quickly, again, on geologic evolutionary time scales, not like in the next couple decades, I assume. Yeah, so there's definitely, I mean, even now there's I'm sure a big pulse of evolutionary innovation that's happening as new things are trying to keep up, or as existing species are trying to keep up with change. But when we talk about this, we kind of take the overall average view. And so right now we're in a time of an average loss of species. And when that ends, we'll be back in a time of average increase in species. And so that's what we think about, that total recovery of biodiversity and not individual species. That's what we're referring to. But yeah, I'm sure that evolution is happening right now.
Yeah, I think it would be quite exciting if we get to see anything new coming out of all this destruction. So Antonio Cruz is asking a little bit more about the kind of soil and ground in the area where the meteorite impacted. You mentioned before that the type of soil in the Yucatan Peninsula had an effect. So he's asking how aerosols and sulfates liberated in the zone had an effect in compounding the impact. Yeah,
this is one of the really cool things about the Cretaceous mass extinction, that it's a really fun what if, because the Yucatan is a carbonate platform. It was a shallow water carbonate platform, basically like the Bahamas today, probably with less islands, but a shallow water carbonate platform, which would have had a lot of lagoons and little enclosed areas that would have been evaporative, which would have concentrated seawater and formed these salts and these anhydrites that have a lot of sulfur in them. And we know from cores taken outside of the crater that these anhydrite layers are relatively thick. A large percentage of the overall thickness of the carbonate system in the Yucatan from the Cretaceous includes these anhydrites. And when we look at the cores that we've taken from the crater, particularly from the peak ring, when we look at the breccia that we see lots of representation of the other types of rock that existed there in the Cretaceous. What we don't see is any anhydrite clasts, any little pebbles of anhydrite that were reworked from the impact event, which suggests to us that most of this stuff, maybe all of it, was vaporized. Maybe it was easier to vaporize anhydrite than carbonate or something along those lines. But this stuff was mostly vaporized and put up in the atmosphere. And this is probably the main cause of the extinction, particularly in the oceans, that it blocked out the sun and caused all this cooling. If you don't have that, I don't think that you have this shutdown of photosynthesis, and I don't think you have a big extinction event in the oceans. Maybe you don't even have a big extinction event everywhere on land. Maybe North and South America, you've got all these wildfires, and you have a local extinction of all the species that lived on those continents. But maybe dinosaurs in Africa and Asia are doing fine. and continue on. Which means that if the asteroid had hit in a different place, maybe we wouldn't be having this conversation right now because mammals never would have taken over the world, right? Which is just fun to think about, that if there's this space rock that has been careening around the solar system for hundreds of millions of years, 22 times faster than a high-powered rifle bullet, and it's coming in and it hits the Earth at exactly the right time to hit right in exactly the right spot to cause this extinction event. And if it had come in 2 minutes later and crashed into the Pacific Ocean, it wouldn't have done anything, or not done anything as severe, which is just amazing to think about.
Thank you.
Yeah.
Blanca Morales, I think this idea of a new mass extinction coming generated a lot of interest and concern. So Blanca Morales is asking, what are the indicators that we have already arrived to a point of no return in terms of extinction events?
Well, I don't think that we necessarily have arrived at a point of no return. I think that's the key thing to think about here, is that it's definitely within our power to save a lot of species and prevent their extinction. And that, I mean, that goes from like charismatic species like the rhinoceros that people are trying to breed and have stick around, or people are protecting elephants, but also fish species and insects and clams and everything else. These things are extinction is being caused by climate change, of course, overhunting, overfishing, pollution, ocean acidification, so buildup of CO2 in the oceans causing drop of pH, and habitat fragmentation is another really big one, where you have a large patch of jungle, say, that can support a jaguar, and then you build a bunch of farms and you fragment that jungle, and suddenly it can't support a jaguar anymore, and that particular jaguar dies, and so you have a fragmentation of this ecosystem that suddenly can't support as large or as many animals. So those are all the things that are causing this. And so it's definitely within our power to stop doing those things, to enact policies, to incentivize people to stop polluting the oceans, to stop destroying habitat, to switch to a non-carbon-based economy. I mean, these are the things that are going to stop this. And if we keep doing the things that we're doing, then we're on that path. But I definitely would not say that we have crossed the point of no return. I worry about it, and it's possible that we have, at least for some species, certainly for some species. I mean, things are going extinct every year. And so there's some species, as we're talking right now, that are still alive, but there's nothing we can do to save them in the next couple of years. But overall, in the sense of there being another extinction event, I think we still very much can affect the course of things.
Yes. So Julio Salvide is asking whether unusual sun activity can have an effect on biodiversity and trigger extinctions as well. I guess before the actual sun just starts to dwindle.
Oh yeah.
But
right,
variations in sun activity, whether they have any effect.
That's a good question. I don't think so. I mean, so in the sense of like the last couple thousand years, when you look at sunspot activity or things like that, it causes, tenths of a degree of climate change, but nothing really major. Another interesting idea would be, what effect does it have on the ozone layer? And that's maybe not so much, well, actually, yeah. So the solar wind does have an effect on the ozone layer. And so that might be part of it too. There's some idea that the Chicxulub impact may have also, as it completely screwed up the entire Earth system, affected the atmosphere in a way that it took out the ozone layer for a while, which would again have probably a pretty negative impact on anything that lived on the surface. But I don't know that sun activity has been directly tied to any extinction events in Earth's past.
Thank you. So I think we've gone long enough already, so we're going to bring down to a close all the questions. I would just like to get a few thoughts on how is it that you chose to do what you're doing, and how is it that you got involved into this huge multinational, super expensive project in the first place?
Yeah. How did I get into what I was doing? I mean, into micropaleontology in general. I chose geology as a major in college because I like to be outside, and I wanted something where I could be outside part of the time. I was really close to going to Virginia Tech and studying forestry. And so I wound up going to Mary Wash instead, and I studied geology. And even there, I was deciding between environmental science and geology. And environmental science had a lot of like biology and chemistry in it. And I was like, I don't really want to do that. And my intro geology course was really cool. I got really sucked into like Earth history stuff, and I got really excited about it. And then when I was an upper level student, I did well on a SED Strat course, and the professor asked me if I wanted to do an independent study. And he was a micropaleontologist working on ostracods. And so I said, yeah, sounds great. And that got me hooked on micropaleo. And I went to grad school for it, and started working on forams, which was awesome. Got really excited about that. I defended my PhD in 2015 in April. Around that time, I think it was like 2 weeks after I defended, was the call for applications for Expedition 364, which is the one that drilled the crater. And I already had a postdoc lined up, and I was like, should I apply? What the heck? I'll just throw my hat in the ring. We'll see what happens. The really great thing about scientific ocean drilling, about IODP, is that they are really good about taking people at all career levels. So it's not just really distinguished scientists that get invited to go on these things. They also look for graduate students, they look for postdocs. And so I'd actually never studied the Paleocene before. And they were like, but I still got this opportunity to sail and go offshore and do all this work. And so, I mean, the answer to how did I get involved in this kind of work was basically just, I started doing something I thought was cool and interesting, and I took advantage of opportunities as they came along. And I've been very lucky in that things have worked out really well so far. But I kind of pinch myself, because like studying how the dinosaurs would extinct is like a 5-year-old's idea of what geologists do, and I actually get to do that. And that's really, really cool. So.
Wow, yeah, I think there will be lots of people who are inspired by that. Thank you very much for that excellent presentation. I think we all learned a lot. And I like that you gave a word of warning of actually what we're looking that happened to the dinosaurs could easily happen to us. And unlike them, we would be the cause. So thank you very much. And also thank you to Ligia for lining up this fantastic speaker. I also want to thank Dr. Gerardo Arizmendi for standing up for Dr. Martín Serrano. So a big hug to Martín and Kenya, and I hope tomorrow looks a little bit brighter than today. So we have some hellos and congratulations from a number of people from UNAM, from Ecotec Desarrollo Sustentable, from several places in Mexico, including Yucatan and Puebla and Guanajuato, as well as people from Argentina. So to everybody who's watching us and joining us today, thank you very much. And I also want to give a special thanks to Professor Matthew Wills, who joined us today. I hope that you enjoyed that talk as much as I did. He is also an expert on evolution and occupation of the morphospace. So I think a lot of his work is quite relevant. So I am going to pass. One other thing. I would like to remind everybody that we have another talk as part of the cycle of seminars from the Universities for Science Consortium. next week on the 14th of July, same time, so that's 12 o'clock Central Mexico time and 6 p.m. UK time. Dr. Victor Sanchez from UNAM is going to talk to us about migration in monarch butterflies. So that talk is going to be in Spanish. So to all of those who are fluent in Spanish and or would like to practice a bit more, we would be very thankful if you join us. So I'm going to pass the word back to Dr. Jaime Urrutia. Thank you.
Muchas gracias. Thanks very much, Chris. Very interesting presentation. And Chicxulub is a major event in life evolution. And great you joined the drilling expedition in Yucatan. This is just great. And we look forward to continue with the studies. Thanks very much. Thanks to all the institutions making this a success. The University for Science Consortium has been a major initiative, and we are very happy with the outcome. We invite to the next presentation on the migration of the monarch butterfly from all the way from Canada, the US, and Mexico. So that will be really interesting. And muchas gracias a todos los que nos acompañaron de diferentes partes. Y le pasamos la palabra a nuestro presidente de Fundación UNAM, Dionisio, licenciado Dionisio Meade. No sé si quiera comentar algunas palabras. Y muchas gracias. Thanks, Chris. And we look forward for meeting you here or in Austin.
Yes. Yeah, thank you guys so much for having me. I really enjoyed it.
Muchas gracias. Muchas gracias a todos. Muchas gracias a Christopher por su excelente presentación. Yo quisiera pues hacer unas muy breves reflexiones. Cuando él habla de la extinción, de la sexta extinción masiva, yo creo que esta pandemia nos ha dejado muchas lecciones. Subrayo dos principales. Una primera es que hay mucha más conciencia del cuidado del planeta. Hoy todo el mundo estamos atentos a qué pasa, a cómo podemos contribuir, a cómo lo podemos mejorar, a cómo podemos evitar que vuelva a suceder una cosa como estas, y eso me genera optimismo de que no llegue esta sexta extinción. Yo creo que con las lecciones que la humanidad ha aprendido, pues hemos aprendido lecciones también para evitar que esto vuelva a ocurrir. Y creo que la pandemia nos deja también en estas etapas de recuperación algunas de las etapas también que aquí se señalan en otros momentos. Una de corto plazo, que es quizá la que estamos enfrentando ahora, una de mediano plazo, cuando podamos disponer de unas medidas terapéuticas, y una de largo plazo, cuando contemos con la vacuna. Yo creo que en esta parte de la lucha por la salud, pues nos enfrentaremos también a estas etapas. Pero creo que la segunda lección, después de cuidar el planeta, es que estos espacios de comunicación a distancia llegaron para quedarse. Cardiff, Gales, Bath, la Universidad de Texas ahora, la Universidad Nacional la próxima semana, la presencia del Colegio Nacional, de la Universidad de las Américas, la propia nuestra, señalan las capacidades y la fortaleza que deriva de un consorcio como estos. Creemos que esto es solo el principio. Y en otra expresión, lo que hemos venido haciendo en nuestras otras pláticas, un recorrido por Cuatro Ciénegas, un recorrido viendo la importancia de los murciélagos, de los recorridos en nuestro barco Justo Sierra, alrededor de Chicxulub, lo próximo que tendremos de las ondas gravitacionales, son solo una muestra de lo que hoy podemos esperar como este espacio de comunicación y de cooperación entre universidades. Seguramente volverán las clases presenciales, pero lo que formará parte de nuestra nueva realidad será esta comunicación a favor de la ciencia, a favor de la mejora de la humanidad a través de estos espacios de comunicación a distancia. Gracias otra vez a nombre de Fundación UNAM. Gracias, Jaime, por habernos abierto el espacio para poder trabajar juntos en esta iniciativa. Y esperamos ilusionados continuar con este programa, ahora en vuelo con las mariposas monarca. Muy buenas tardes a todos y gracias de nuevo.
Muchas gracias a nuestro presidente de Fundación UNAM. La doctora Pérez Cruz quiere hacer unas palabras.
Solamente quiero agradecer la oportunidad que nos dieron para la presentación de Chris, tanto al Consorcio de las Universidades como también a Fundación UNAM y al Colegio Nacional. I say thanks very much to the consortium, to the Fundación UNAM, and also to the Colegio Nacional to give us the opportunity that you can give this interesting talk about not only about the extinctions, you include a lot of topics about the importance of the drillings for sciences and for everything, and the role of the human being in this earth. Thank you, this planet. Thank you very much, Chris, and thanks very much to everyone. Thanks.
Okay. Well, thanks very much, and thanks again for your interest in this cycle of conferences. Thanks, and thanks, Chris, and see you soon. See you everybody soon.
Yeah.